was individualized but related to the starting microbiota diversity that these participants had. Okay, let's shift over now to the fermented food group. So this was a group in this arm, participants didn't have to change anything in their diet other than incorporating as much fermented foods as possible. They ate all kinds of stuff, kimchi, sauerkraut, yogurt, kombucha. They could do whatever they wanted for fermented food. Unlike the high fiber diet group, the fermented food arm, their nutrient profile didn't really change much. The only difference that we saw was an increase in the amount of animal protein they consumed. And this came from fermented dairy foods like yogurt that they were eating more of. We were concerned that perhaps participants' sodium intake was going to go up. A lot of fermented vegetables have a lot of salt in them, but in fact, we didn't see that. So as participants were consuming more salty fermented foods, they must have been cutting it out in other aspects of their diet, probably without even realizing it. Okay, what happens now to the microbiota of participants consuming fermented food? We saw a dramatic cohort-wide increase in microbiota diversity. These participants at the beginning of the study had about 100 different species that we could detect in their gut. By the end of the study, they had over 125 species. So they gained on average another 25% of microbiota diversity on top of where they started. And at first, when we saw these data, we thought, well, I guess that makes sense. I mean, they're eating bacteria, right? Fermented foods have live microbes in them. So this increase in species that we're detecting is just the bacteria that these people are consuming. And so we wanted to test that directly. So what we did was went out to the grocery store. We had food logs from all these participants, so we knew exactly what they were eating. We went out to the grocery store and we bought all the same foods that these people had consumed. We then struck these foods out onto these agar plates to see if there was actually live bacteria found within these foods. And turns out there were in every food. So when that label says live and active microbes, they mean it. There were live and active microbes in every fermented food that we tested. Then we went and identified which each of these strains of microbes that we detected in the fermented foods, what those microbes were. And this is the list that we have here in the middle of the different bacteria that we found in these foods. And for the most part, the microbes that we found in the foods were the microbes that were listed on the label. So in general, these fermented foods have live and active microbes, like they say, and are largely what is written on the label. So it turns out these are actually pretty high quality products. Okay, so what the 25 on average new species that we detected in these participants' microbiota, how many of those are the bacteria from the fermented food? When we looked at that, it turns out that this small sliver here, which is about 5% of the new species that we detected within the gut of these participants, came from the foods that they consumed. 95%, this whole purple part here, not from fermented foods. So it appears that the consumption of fermented foods is having an indirect effect on microbiota diversity. There's something about eating fermented foods that makes the microbiota receptive to new species. We don't know where these species came from, likely other parts of these people's environment, maybe from their homes, maybe from other people they're interacting with, but it didn't come from the fermented food. Okay, what about these participants' inflammation and immune system? In the fermented food group, we actually saw a cohort-wide decrease in many inflammatory cytokines. So here I have, I think, about 18 inflammatory cytokines. And over the course of the intervention, all these inflammatory cytokines decreased for the whole group. So unlike the fiber arm that had a more individualized response to the immune system, fermented food seems to have a powerful group-wide effect on decreasing inflammatory cytokines. So we learned that fermented foods increases microbiome diversity in participants and decreases inflammation. Now, every time I present this, people ask me, what about probiotics? Because fermented foods contain live bacteria. What if I don't like fermented foods or it's a pain to have them around the house? I just want to take a probiotic that has the live microbes. Isn't that going to be the same thing? So we wanted to test that directly. So this is another human intervention trial that we did. And in this one, we actually recruited participants with metabolic syndrome. We wanted to up the ante. We wanted to see not only can we change these participants' microbiome and immune system, but can we actually change the trajectory of their metabolic syndrome? This was a double-blind placebo-controlled study. So 14 of the participants were randomly assigned to a placebo group, and 28 took a probiotic twice a day for 10 weeks. We have baseline, intervention, time points, washout, blood, stool, so we can measure their microbiota and their immune system during this intervention, and ask the question, can probiotics improve participants' microbiome, decrease their inflammation, and reduce the severity of their metabolic syndrome? Okay, so we had high hopes for the probiotic because the fermented food did increase microbiome diversity. But in this study, we did not see an increase in microbiome diversity with probiotic consumption. So if you look, the probiotic in purple, the placebo in gray, it's all the same from baseline to the end of the study. So no change in microbiome diversity with the probiotics. What about inflammatory cytokines that we saw decrease so dramatically in the fermented food? Did that work for probiotics? And the answer, unfortunately, was no. No difference between the probiotic or the placebo group, and also no difference over the course of the study either. So inflammatory cytokines stayed the same throughout the study. Okay, what about markers of their metabolic syndrome? We looked at insulin, glucose, lipids, all those things unchanged during the study. So at this point, this was a pretty depressing point in the study. We had done this intervention. It was complicated. It was expensive. We were hoping to see changes in the microbiota or immune system or anything. We were seeing absolutely nothing. And so we thought, okay, before we just write up this negative result, let's take a closer look at individual participants and see if there's any information we can get from that. And so here what I'm showing in this heat map is each participant is a row here. And so this is the placebo group. This is the group that took the probiotic. And then each column is a different measurement we took of their sort of metabolic syndrome symptom. And what we did was basically feed this into a computer and ask the computer, do all the probiotic participants look the same? Or do you see anything, any groupings within the probiotic group that are different from one another? And what the computer spit out was actually two different groups that it identified. In one group, shown here in the middle, there was really not much of a change. So that's indicated by the sort of white and light blue in most of their markers. But in some cases, there were actually worsening markers. So worsening triglycerides here and insulin here. That's when it goes red. But in this group down here, we actually saw improvements. So that's indicated by the dark blue. And these were statistically significant improvements in their triglycerides and their insulin. And so we termed this group responders. So these were people that seemed to be getting a benefit from the probiotic intervention. And so we asked the question, why? Why are these people benefiting from the probiotic intervention? And the first place we looked is their baseline microbiota composition wasn't different. Baseline microbiota diversity wasn't different. What about their immune status? Maybe there was something about their immune system that was different at baseline between these two groups. No difference. At this point, we remembered that in addition to this intervention, we asked participants to keep food logs, write down what you're eating throughout the course of the experiment. And when we went back and analyzed these food logs, what we found was that the responders actually had a diet that was different from that of the non-responders. And specifically, these responders were eating more vitamins and minerals and had a slight increase in their dietary fiber consumption. But in general, what we observed was that the responders were eating more plants in their diet than the non-responders. So this was an indication that to gain the full benefit of this probiotic, you had to be eating a diet that was more rich in plant complex carbohydrates. So what these studies taught us is that fiber and fermented foods do different things to our microbiota and our immune system. So if we go back to our broccoli example, when you eat a high fiber diet, those complex carbohydrates make it to the colon where the majority of our gut bacteria live, and these gut bacteria consume these complex carbohydrates. That resulted in participants having a denser community of microbes within their gut, better function of those microbes, more capacity for plant carbohydrate degradation. And we also saw lower inflammation if participants had a higher baseline microbiota diversity. In the fermented food group, what we found was that the consumption of fermented foods led to a higher diversity of their microbiota and lower inflammation. And for probiotics, what we found was that probiotics appeared to work best in the context of a better diet.